Thank you for that introduction, and I am very pleased to present to you the following presentation on the innate immune modulation, reducing F2-isoprostane and improving mental health following acute or chronic stressors. And before we get into the data that I'd like to present, I want to go over a couple key introductory points. First being urinary F2-isoprostane is the gold standard in vivo biomarker for systemic oxidative stress. So changes in this biomarker do reflect changes in the oxidative stress status of the individual or organism. Oxidative stress is defined as an imbalance between the production of reactive oxygen species and the antioxidant defenses of an organism or cell. When you have elevated oxidative stress, it actually paves the way for neurodegeneration diseases and even mental health disorders. And on the right side of this graph, it's just a wheel depicting oxidative stress in the center and all of the potential ways that it can influence mental health or neural function of an individual or organism. Lastly, oxidative stress is associated with aberrant activation of the innate immune system. So one of the sources of the ROS influencing oxidative stress is via the innate immune system. To date, unfortunately, there are very limited treatments to manage oxidative stress, and all of these treatments are focused on exogenous compounds, such as antioxidants, vitamins, polyphenols, and so forth. But the issue you have when you focus on exogenous compounds is that you are not actually addressing the endogenous origins of the ROS. And so this led us to a hypothesis that if you could modulate the innate immune system using receptor agonists, you could actually manage oxidative stress endogenously. But with that introduction, I want to start getting into the studies that I'd like to present in this presentation. The first one being a pilot study where we looked at immune modulation and its effect on F2-isoprostanes and the psychological health of a stress cohort. The treatment in the study was a product ingredient called Reset. It's comprised of agonists for the TLR and NOD2 receptors of the innate immune system. And the receptors, the agonists for these receptors that were contained in the treatment were for TLR2, TLR4, and NOD2. The protocol that was used in this study is depicted on the left side of the slide. You see the inclusion and exclusion criteria for the participants, and this were the participants were all PTSD veterans. The treatment was 24 milligrams taken sublingually, and this was taken twice a day or BID. We monitored the biomarker F2-isoprostane in the first morning urine on day 1, 15, and 70, and then did self-rating psychological assessments also on days 1, 15, and 70. And we used 4 different validated tools. The result on the change in the urinary F2-isoprostane biomarker is depicted on this slide. And what you can see is that we got a significant reduction in F2-isoprostane measured on days 15 and 70. And this rate and extent of reduction in F2-isoprostane has not been reported prior. So we were quite pleased to obtain this result. And when you lowered the oxidative stress, we then looked at how did this influence the psychological health of the participants. And as I mentioned, we used 4 validated tools that the participants did self-assessment on, and this was done on days 1, 15, and 70. And what you see here are the changes in the scoring by the participants over time. And it's interesting to note that the change in improvement of the scores correlates with the reduction in the F2-isoprostane in the urine and consequently reduction of oxidative stress of the individual. And this correlation is quite strong, and the coefficient is recorded and shown in the lower right corner of each of the graphs depicting the psychological tool. Further, we did get significant improvement on the satisfaction with life at day 70, also with improvement in the sleepiness scale at day 70, and then on depression at day 15. So what you saw from this study is two things. First, that you could reduce oxidative stress in individuals by manipulation of the innate immune system. And secondly, the reduction in oxidative stress did result in improvement on the mental health or psychological health of the participants. So having that data in hand, we then wanted to look more in depth at what is going on when you change oxidative stress. And for this, we used the animal model Drosophila, and this was picked for several reasons. First, it has been widely used to study both neural and immune pathways. There is good conservation of neural and immune structures across Drosophila and humans. Methods are available already to assess chronic stresses and acute stresses in Drosophila. And lastly, Drosophila locomotor function is a real-time assessment of neural function. So let's start looking at the studies that we did on the Drosophila or flies. The first one was looking at longevity enhancement. And first, I want to just comment that aging is the accumulation of stress over time. So if you lower oxidative stress in an organism, you should see an improvement in aging. And that is exactly what we did see with this study. You see that we got significant improvement of lifespan up to 23%, which is quite noteworthy because getting lifespan expansion at that level is not common in wild type organisms. Secondly, we also wanted to look at the neural health or motor function of the flies as they age on treatments. So in this study, we are looking at the climbing behavior of the flies, and we started with flies of 1 week of age and then assigned them to treatments, control, and different dosages of reset. And then we assessed their climbing behavior at weekly intervals, starting with week 2 and going through week 5. And what you can see is that starting with dosages that are diluted 1 to 100 and higher, you see significant improvement in the climbing behavior of these flies throughout all intervals of assessment. And when you see improved climbing behavior in flies, what you are actually noting is improved neural function or health of these flies. So these flies were actually maintaining a more healthy neural function throughout aging compared to their control fly treatment group. Next, we wanted to look at if you treat these flies at different ages, does it change the response that you get from the treatment? And we looked at 2 different ages. One was a study with 1-week-old flies. The second study was with 3-week-old flies. And it was the same protocol. You started with the aged flies, applied them to the treatments, and assessed climbing behavior after 1 week. And what we saw was that the treatment reset significantly improved locomotor function over controls, regardless at what age the treatment started. So if they started at an age 1-week fly or 3-week fly, we got the same response. Secondly, it's interesting to note that flies treated with reset for 1 week actually maintained locomotor function similar or better than the pre-treated younger fly that was started at the beginning of the study. So we got essentially neural climbing behavior or neural health of these treated flies that compare to a younger fly. What might be going on in these flies could be related to neuroprotein aggregates that accumulate with age in neural tissue. This is a common phenomenon that is known in organisms. And so what we did is we looked at the neuroprotein aggregates of these flies when they are being treated. And in the bar graph to the right, you see that when we treated the flies for 1 week, we did not get a significant difference in the protein aggregates in the neural tissue. However, when we treated the flies for 3 weeks, we did get a significant reduction in these protein aggregates. So it does suggest that the reset is able to maintain a healthier neural tissue in these flies. We're moving now to protocols where we looked at acute stress. So we're kind of moving out of the chronic stress into treatment effects on acute stress. And the acute stress we used here was a TBI. And this particular TBI that was used in the protocol is a mild repetitive. It was 3 TBIs given in succession. And we looked at 2 different treatment protocols. One was the treatment was started 2 days prior to the TBI and then maintained for 1 week before we did the climbing assessment. And the second treatment protocol was a post-treatment where the flies were put on treatment immediately after the TBI concussion, and then the climbing behavior was assessed 1 week after the TBI. And what you see in both of these graphs is very similar, is that the treatment significantly improved the locomotor function after the TBI. So you see, and that is compared to the TBI control. Interesting also is the treatment resulted in locomotor function similar to the non-TBI control. So these treated flies behaved essentially the same as a fly that had not been concussed. And the treatment effect was similar regardless of how the treatment was applied, either pre or post concussion. And we know that when you do an acute trauma to the head or neural tissue, you induce a inflammation response. And what I have here is the innate immune system cascade. And I wanted to just quickly go over this because it'll help with the gene expression work that I want to report on next. So up in the upper left corner, you see the TLRs are there showing the apex receptors of the cascade. They funnel into a key point of NF-kappa beta, and this is a protein that is a DNA transcription factor. And the NF-kappa beta protein does elicit the downstream products produced by the cascade and which products are produced. And the one that we monitored in our studies was the antimicrobial peptides, or AMPs. And so the next data that I want to talk about is gene expression profiles of both NF-kappa beta and AMPs examined of the control and treated flies. When we look at the NF-kappa beta gene expression, so this is the left side of the graph or the slide here, we notice several things. First, the expression pattern following the trauma was normal. There was no difference in terms of the expression pattern across control or treatment. However, there were 3 key points different of the treated flies versus the control. First, the resting state or basal inflammation level of the flies was lower in the treated flies compared to the control flies prior to the TBI. The gene activation after the TBI was stronger and more rapid in the treated group. And also the resolution of the gene activation was more rapid and complete in the treated group. And this is very important because what you want is you want the inflammation response, but you also want it to resolve very quickly because that prevents chronic inflammation due to the trauma. And a good way to depict this resolution or reduction in the basal inflammation is with this bar graph looking at the AMP expressions. And you see that the 3 AMPs that we measured were actually significantly lower in the treated flies than the control. So this last slide that I want to present on data is preliminary data. And what we looked at was tau in the Drosophila. And this was transgenic Drosophila where the human tau gene was inserted. And we did this because we wanted to understand, does tau behave similar to treatment as the protein aggregates that we showed earlier in the presentation. And what you see here in this bar graph is the answer to that question is yes. In flies transgenic flies that were treated with Reset for 1 week, we saw that the total tau in the neural tissue was significantly lower. The phosphorylated tau in the neural tissue was also significantly lower. So that was actually very encouraging for us to see. And what this tells us is, in summary, that endogenous management of oxidative stress via the innate immune system can be a new tool for neural health goals. And I just want to quickly refresh and summarize the data that we saw today. So first off, we saw reduction in oxidative stress was demonstrated by the reduction in urinary F2-isoprostane levels. And this reduction in oxidative stress then resulted in improvement in mental health, extension of lifespan, improvement in neural function across aging, improvement of neural function regardless of when treatment was applied, improvement of neural function after an acute stress such as TBI, that if you reduce oxidative stress, you also reduce basal inflammation levels in an organism, you reduce neuroprotein aggregates, and you also reduce a tau protein in neurotissue. So where this takes us now in our research is that we want to continue to look at reduction in oxidative stress and its effect on neurotissue protein aggregates and tau, and how that feeds into neurodegeneration and ultimately into Alzheimer's. And with that, I am going to stop talking and open the floor up for questions. And if you do want to contact me about this presentation, please reach out to me at the contact information provided on this slide. Thank you.